Good evening, Manchika. I'm Rahul Saha, and today we'll be discussing about the later part of the same chapter, organisms and populations. So the last time we had talked about the abiotic factors, the biotic factors, the biome, the ecosystem, the biotic community, and we had just commenced our talk regarding the atmosphere. So we all know that in case of atmosphere, the main good evening, Ayman. So the main thing which governs our body is oxygen. And why do we need oxygen in the first place? The answer of that question lies in class 11th. If you have been taught properly, then you would be knowing that in the ETS chain, you have something known as complex 4. So from cytochrome C complex to oxygen, the transfer of electrons take place, and oxygen is the best acceptor of electron. If this does not happen, you wouldn't be transporting out H plus into the intermembrane space, and then you wouldn't be generating the chemiosmotic gradient. Or chemiosmotic theory is there. The gradient is known as electrochemical gradient. And if you cannot generate electrochemical gradient, the H plus will not revert or will not return into the mitochondrial matrix via the complex 5, which is the F1, F0 ATPase. And therefore phosphorylation of ADP cannot occur. ATP formation cannot occur via binding change mechanism. There are various mechanisms, rotation mechanism, binding change mechanism. Okay. So this O2 is extremely essential. So this is the only reason why we need oxygen. If there was any other gas which could accept electrons or have more affinity towards electrons than oxygen, then would be soluble in blood as per the Henry's law, which you might have learned in chemistry, right? That solubility is equivalent to, you can say, the partial pressure. So P equal to, you can say, KHX. That is the Henry's law. Okay, and then also binding affinity with hemoglobin. So it should bind with hemoglobin so that hemoglobin can carry it and deliver it to the tissues. So if these three factors or these three criteria were satisfied, then we could have breathed in any other gas, and the fourth criteria is it should have been readily available in air, right? You cannot just purchase it. Good evening, Ayushi. Good evening, Tarun. So these are the reasons why we need oxygen in the first place. Okay. So now coming on to the second factor, which is light or the radiation factor. Again coming to chemistry and physics. So yesterday I would like to give you some specific information, since you are all science students, that yesterday we had something known as the biological Nobel Prize. So Nobel Prize in biology was, you can say, bestowed upon three major scientists. Okay. So the three scientists are, you can say, the number one scientist is Harvey J. Alter. So yesterday the Nobel Prize in biology in 2020, the category physiology or medicine, was awarded. Okay. So you are fond biologists, so you should be knowing this. So the three scientists are Harvey J. Alter. Okay. He presently works at National Institute of Health, California, USA. Then Michael Houghton. Okay. Show you. He presently works at the University of Alberta in Canada. And last but not the least, Charles M. Rice. He works at the University of Rockefeller, New York, USA. So these three scientists, they have contributed immensely and profoundly towards the discovery of hepatitis C virus. Hepatitis C. Okay. Because hepatitis B is easily recognizable, hepatitis B was not recognizable, and there was no test to recognize hepatitis C. And hepatitis C can also cause liver cirrhosis, which is an irreversible liver damage, fibrosis of the liver, and then that could lead to hepatocellular carcinoma also, or liver cancer. So now these three scientists, they have worked independently to develop first testing of hepatitis C. If you have got hepatitis C, it could be a silent killer also, and then antiviral treatments, and now you can be cured of hepatitis C too. Okay, so because of these specific achievements, they have been bestowed with the most prestigious award. It is the Nobel Prize in the field of physiology or medicine 2020. Specifically, this scientist, Charles M. Rice, he works at Rockefeller, and my previous guide at VIT University, Dr. Everett Nelson, so he used to work in Rockefeller University as a postdoctoral fellow. So he called me up yesterday. So he was telling that he worked with, you can say, another scientist, okay, known by the name Dr. Barry S. Scholar. And Barry S. Scholar and Charles M. Rice, they are, you can say, nowadays they are forming a collaboration, and they both are working against SARS-CoV-2. So these three are, you can say, all these three scientists are professional virologists, right? So that is the thing here. So, and today we have got Nobel Prize in, you can say, physics. Nobel Prize in physics has been delivered today, okay, to black hole formation, to three scientists who were responsible for black hole formation, okay, who discovered it. One was Sir Roger Penrose. He is 89 years old, okay. Tomorrow will be Nobel Prize of chemistry, okay, distribution. Roger Penrose, who works at University of Cambridge, not Cambridge, University of Oxford, you can say, in UK, and he had discovered that black hole formation is a robust prediction of the general theory of relativity proposed by Albert Einstein. And then two more scientists, Professor Reinhard Genzel, okay, Genzel, and one women scientist, okay, one woman scientist, Professor Andrea Miages, okay. So being a woman, she is a great physicist. So these two scientists, Professor Reinhard Genzel works at University of California, Berkeley, and also Max Planck Institute for Extraterrestrial Physics in Germany, and Andrea Miages works at University of California, Los Angeles, UCLA. So these two scientists also were awarded for their discovery that a supermassive compact object like the black hole only is present at the center of our galaxy. So this was the Nobel Prize in Physics, which was awarded 2020 Physics Nobel Prize. Okay. So these two were the, you can say, top news. Now coming to our light as a factor. So light, we know that has got both particle and wave nature via quantum mechanics. We know that. So light is a very important factor for climatic control, right? We have photoperiodism, right? So differentiation of plants, growth, development is directly affected by the light, okay? And it's a very, very indispensable factor for vision. So cornea size, eye size, again skin pigmentation. So people living towards the equator, like the African one, they are darker because they have more exposure to light, so more melanin synthesis. Okay, melanin has also got anti-cancer properties. Okay, then reproduction, then biological periodicity of animals. So that all also depends on light only. So light is a great factor here. So if we analyze each factor of light that comes in your board examination, so how does color vision? So first of all, I'll take color vision as a factor which has been affected by light. So it's the ability to differentiate different colors, okay, and is well marked in fish, birds, primates due to presence of different kinds of, you can say, cone cells. Okay, you can have erythrolap cone cell, you can have cyanolap cone cell, you can have chlorolap cone cell. in the retina of their eyes. Okay, there are various kinds of cones which can discern different types of light. Okay, different intensity of light. You can have erythrolapse, you can have cyanolapse. Okay, then you can have chlorolapse. Okay, there are many are there. In, you can say there is a specific animal you might be knowing, salamander, Proteus anguinus, that is completely eyeless. And sharks you might be knowing, great white and all, tiger shark, hammerhead. So sharks are color blind. They cannot discern or recognize color. Okay, they are color blind completely. Okay. So, and if you talk about the size of the eye, so marine animals which live at a depth of 500 to 3000 meters have, you can say 3000 meter depth, I should be writing, depth, have larger eyes because light penetration is pretty low there. Okay, so larger eyes are needed, more cone cells, more better cornea. Okay, more light, more, or you can say larger aperture. Okay, then coming on to skin pigmentation as a consequence of light. Skin pigmentation. So light intensity induces chemical changes. We all know that, which leads to pigment formation in the cells. So you might have noticed tanning. Tanning is what, even like, I can talk about myself when I used to be, you can say, of your age, I wasn't tanned. But when I switched to college and also, my college, the entire study happened in South India, VIT Vellore, then IIT Chennai, IIT Madras originally. It is also it is still known by the name IIT Madras, even though Madras has been converted to Chennai. Okay, like IIT Bombay. It is not IIT Mumbai. That name hasn't changed. So tanning. So the synthesis of melanin is too much. Okay, in the chromatophores or melanocytes, you can say that is tanning. Okay, so my hands have got tanned and it has not been reversed. Okay, till date. So face color and my hand color are completely different because of tanning. Okay, in South India. So this occurs due to too much melanin formation by, you can say, chromatophores, color, or melanocytes, which are the color producing cells. Okay, too much formation of melanin. So this occurs in response to prolonged exposure to bright sunlight. I told you that melanin has anti-cancerous property. So it acts as defense against UV radiation or sunburn. Okay. Now we had talked about photoperiodism. That is again a consequence of light. Photoperiodism. Okay. So what does it mean? By definition it is asked. Okay. So I am giving you the definition. It refers to the response of organisms to the duration of sunlight okay, in a day, or simply known as photoperiod. So how much light are you exposed to for how many hours? So what is the time duration of light that you are exposing yourself to? Okay, that is known as photoperiodism. So in many animals, migration, reproductive behavior, foraging are controlled by relative lengths of day and light. Okay. So if you talk about especially the reproductive activities, then these are nothing but the photo period. Okay, means the time duration for which you are exposed to the light is directly known to affect the breeding behavior of certain insects, certain birds, also certain mammals. Okay, for example, long photoperiod of late spring and early summer. Okay, long photoperiod of late spring and early summer. Okay, what it does is it stimulates the secretion of neurohormones and production of eggs in the birds. The secretion of neurohormones. Okay, which will lead to production of eggs or ovum in the birds. Ovum in birds. So on this basis, animals are again divided into three categories. Okay, like you have short day plants, you have short day animals, you have day neutral plants, you have day neutral animals also. And then like you have long day plants in case of botany, you have long day animals also. Okay, that's a pretty enthralling thing, but it exists. Okay, so short day animals. Means which breed in decreasing photoperiod, like autumn, breed in autumn season. Okay. Like sheep, deer, less exposure to light. Long day animals, which breed in lengthening photoperiod. Okay. Okay. So, like spring and summer. Okay. The example could be turkeys, starlings, etc. Then you have something like day neutral animals which breed throughout the year. Doesn't matter how much photoperiod they are exposed to. They choose to breed throughout the year. It doesn't affect their basic biochemical metabolism, right? Or their basic metabolic rate in any way that it will hinder their reproductive rate or reproduction rate. So day neutral animals. Okay. So the examples are rabbit, guinea pig, etc. Okay. Now the next thing that light can control is our circadian rhythm. Circadian rhythm means you may have learnt it in class 11th. If you haven't, then those questions will be asked in NEET examination. Circadian rhythm is also known as biological clock. It depends on two things as far as your NEET examination is concerned. One is SCN, known as suprachiasmatic nucleus. Present in the hypothalamus. We have multiple type of nuclei in the brain. Nuclei doesn't mean the nucleus of a cell. Nucleus or nuclei, when it comes to neurology, means the collection or cluster of the cell body or cyton or perikaryon or soma, whatever you want to call the cell body as. There are 4 synonyms for that. So the collection of cell body or the gray matter, okay, in the central nervous system, that is known as nucleus, and same will be known as ganglia or ganglion in PNS. Collection of cell body in the peripheral nervous system is known as ganglia or ganglion, okay, singular. So suprachiasmatic nucleus in hypothalamus And if you talk about epithalamus, okay, so epithalamus region of the brain has got something known as pineal gland. So non-exposure of light, you can say it will stimulate the suprachiasmatic nucleus, which will then stimulate the epithalamus, the pineal gland, okay, to secrete a specific hormone known as melatonin. And melatonin directly puts you to sleep, okay, via a complex signaling cascade that is known as your circadian rhythm. Okay, it puts you to sleep. That's why we sleep at night more. We can sleep at daytime also if you're tired, but usually the less amount of light intensity causes us to drowse, causes us to get into or drowse away or drift away into sleepness or sleep. Okay, under sleep also you have two types of sleep. You have NREM sleep, okay, you have REM sleep. That is rapid eye movement, non-rapid eye movement. This is sound sleep. Okay, this is kind of shallow sleep, you can call it. Okay, shallow sleep. So this also comes in NEET examination. Okay, so I think I taught you about the brain waves. Okay, I remember teaching you or somebody else about the brain waves. So I think in class 11th I taught. So brain waves are also directly asked in NEET examination. Okay, so we have delta wave, we have theta wave, we have alpha wave. Okay, we also have beta waves. Okay, we also have gamma waves. Okay, those are directly perpetuated from the brain and measured via EEG, electroencephalogram. Encephalo means brain. Okay, so electroencephalogram. Okay, so you have something known as delta wave. Okay, so I'll tell you about waves also because they are asked in NEET examination. You might not have read about it in class 11th. So delta waves, when we talk about delta waves, their frequency is 0.5 to 3 hertz. Okay, that is their frequency. So they are, you can say, are very slow brain waves. Okay, low frequency. Okay, they are generated in the deepest meditation and dreamless sleep. Okay, that is the non-REM sleep, when you are sleeping sound, nice sleep you have got, undisturbed. Okay, so they suspend external awareness and are the source of empathy, you can say. Okay, so you can say deep restorative sleep, healing process. Okay, when some psychologist is trying to heal you. Okay, or you can say non-REM sleep, means deep sleep. That is your delta wave. Okay, then theta waves occur also mostly in sleep. So theta wave, the frequency is 3 hertz, okay, to 8 hertz. So theta wave is our gateway to learning, you can say. Okay, in theta our senses are withdrawn from the external world but are focused on signals originating from within. Okay, so you can think of it as the twilight stage. You might have watched the movie. So it is where, suppose I have closed my eyes properly and I am drifting to sleep but I am not really sleeping. That is what your theta wave is all about. Okay, you can call it REM sleep, okay, type of REM sleep, or just you can say towards sleep, not completely, you can say slept. Okay, not completely drowsy fully. Then you have alpha wave. So alpha wave is 8 to 12 hertz. That is our alpha wave. So alpha brain waves are dominant during quietly flowing thoughts and in some, you can say, meditative states also. If you are meditating, then alpha wave will be dominant. Okay, suppose I have just shut my eyes but I am completely awake. Okay, that time it could be awake. Okay, mental coordination, calmness. Okay, that is your alpha wave. Then comes beta waves. Okay. So beta waves are 12 to 38 hertz. Okay. So they dominate our normal waking state. Like when I am teaching you, I am in the beta state. So if you go for my EEG right now, you will see beta waves being predominant. Okay. So it's a fast activity wave. Okay. So it is again further divided into many types. That is not in your course. You have low beta. You can say simple beta, beta 1, beta 2, beta 3, beta 4. Okay. Mostly beta 3 is there, beta 4 is not there. So, but that is again going into deep neurology. It is not in your course. So you can think of it as directly, suppose you are teaching or you are learning, so eyes open, full concentration, that is beta wave. Okay. And then gamma waves are there, which are again not in your course. They are like some people have brains in 10 directions. Okay, their brain is not stable. Not stable in the sense they are not mad, but some people are there. Suppose I am teaching you. You are thinking about some movie. You are also thinking about something else. You are also thinking about something else. You are at 10 places at one go. Okay, you are not able to concentrate properly. Many students have seen they'll say, "Sir, I am at 10 places at one go." Not quite able to concentrate. So that time, there, you can say gamma wave would be more active. Okay, so it's not in your course, but just for the sake I'm telling you, gamma wave is 38 to 42 hertz. Okay, till beta wave it's there in your NEET examination. Okay, so that is beta wave. So that is your biological rhythm, which is controlled by light. That's why we should not disrupt our sleep. So earlier times there was less incidence or less occurrence of cancer because people used to sleep at right period of time or right point in time. Now we don't sleep at right point in time. We have become like night crawlers or night dwellers. Like owls, we have become nocturnal creatures. But we are really a diurnal creature, right? So by doing that, we disrupt our circadian rhythm and we suffer from insomnia, less sleepiness. Then we go to benzodiazepine, you might have learned, tranquilizers. Okay, then we can go for narcotics. Okay, so all that we rely upon at a very tender age, early age, and that disrupts the complete neurochemical or neurological or neurotransmitter balance in our brain. Okay, and then that disrupts mainly the melatonin, and melatonin has been found to be anti-cancerous too. So you can see the correlation that, or just try to perceive it, that you might not have heard your grandparents getting or dying due to cancer, them getting diagnosed with cancer, but nowadays cancer has become very, very, you can say, common thing, a common notion, because the sleep pattern is gone. Because we are too busy, our sleep pattern is gone. It's completely rattled, right? Completely we are in a disarray. That is why cancer states or cancer number has skyrocketed, you can say. Of course, there is medical diagnostic tools, but still it is too much. Okay. So metabolism is also there, one of the points. So light can increase our metabolism also. Okay, light increases the enzymatic activities and general metabolic rate. Enzymatic activity and general metabolic rate. Okay, then locomotion also. You'll be astonished to know it does affect locomotion also. Okay, so the regulation of speed of locomotion by light does happen, and that is known as photokinesis. Kinesis means movement controlled by photon equal to h nu, photokinesis. That there is an example that blind larvae of mussel crab, move faster in higher light intensities that has been seen. So larvae which are blind, okay, of mussel crab, okay, they move faster in higher light intensities, which can be measured in candela or lux. Okay. Higher light intensity of light. The standard unit is candela, one of the 7 basic units in physics also. Okay. And the orientation of animals can also be changed and can also depend upon, you can say, light during locomotion. That is known as phototaxis. That is the orientation of animals during locomotion in response to light as a source, like euglena are positive phototactic. They'll go towards where the light source is. Okay, you can say positive phototactic. Okay. And you have earthworms, slugs, which are negative phototactics. Negative phototactic. Okay. So now we'll switch on to the next thing, that is effects of light on plants. Very, very important topic. That was animals. Now this is plant body. Okay. Now synthesis of pigments, number 1 point. So we know that the synthesis of chlorophyll A, B in the leaves and bright pigments otherwise, okay, like lutein, zeaxanthin, then you have beta carotene, alpha carotene in the flowers and fruits, is controlled by light, and light helps. That light is the, you can say, not only helps, it is the pivotal factor in photosynthesis, which is pH2 680 nm, might have read 700 nm is the PS1. Okay, so 680 nm, PS2, 700 nm, that is PS1. Then you have a concept of PAR, photosynthetically active radiation. Okay, then number and position of chloroplasts. So the upper surface of leaves, again this is a good concept here, it's there in class 11th also. I'll repeat it for you. So number and position should be a good number of position. So number and position of chloroplasts. So the upper surface of leaves of sun, of, you can say, some plants have more chloroplasts. Okay, upper surface of some plants or some leaves have more chloroplasts. Okay, and are arranged in line with the direction of light in order for them to screen. Okay, they need to screen, okay, from the effect of strong light. But in the leaves of shade plants, okay, the chloroplasts are always lesser in number and are arranged at right angle to the direction of light. Here they are arranged along the angle so that you do not have photo bleaching. Okay, too much light will cause too much formation of oxidants, okay, or reactive oxygen species, reactive nitrogen species, like you will be forming superoxide, peroxide, then hydroxyl ions, which will be quenched by the beta carotene and all, which I had explained to you. They are the quenchers, they are the antioxidants, free radical scavengers, but how long, right? because if you look at the sun with your naked eye, you will be blinded within minutes, because we have lutein, zeaxanthin in our eyes also, retina, for quenching, but still the amount is quite less. It cannot provide us, you can say, incessant protection against high intensity light. That's why we are not allowed to look at any kind of lunar or solar eclipse. We need specialized rays. Direct UV radiation will directly cause extreme DNA damage. Okay, DNA repair pathway will be unable to, you can say, fix it. Okay, so the thing is, the shade plants have it in right angle at the direction of light to receive as much light as they want, because they are in shade and they want to screen light. Okay, be protected from it. Okay, so the arrangement of chloroplast is changed. Is they vary. The arrangement of chloroplast, you can say, vary significantly with the change in light intensity. Okay, then again, photoperiodism that we had talked about. We have long day plants like radish, spinac. I'll just give you the example. You know the already the concept. Photoperiodism. We have long day plants. Okay. So again, you can say the angiosperms are divided into three categories mainly. So we have long day plants, that is radish, spinac. Okay. Might have eaten both, right? Then you have sorghum, short day plants. Again, less photo period, long day, long photo period. They are like cosmos, datura, salvia, cannabis. Okay, cannabis is there, cannabinoid receptors are there, right? Cannabis is there in that chapter also. I think chapter 8 only. Then datura, these are short day plants. Okay, LSD and all, lysergic acid diethylamide. Okay, so then you have day neutral plants like cotton, tomato, potato. It doesn't matter how much is the photo period. It doesn't depend upon the amount of time that light is available for them to utilize. Okay, that is very common. Tomato, potato, cotton. You can remember the scientific names also if you want. So that depends upon you. Then we have something known as transpiration also. We are all aware of transpiration. Then transpiration cohesion pull theory. We know guard cells. We know transpiration stomata. Okay. So light will increase the rate of transpiration indirectly. Okay. How? By increasing the temperature of the leaves and promoting the opening of stomata. So it's an indirect effect. Okay. And that will cause water movement from root to shoot via tracheids, vessels, xylem parenchyma and xylem fibers, right? Via the xylem vascular tissue. Again, we have that transpiration cohesion pull theory also, by which the water can proceed upwards against gravity at a slower rate. Okay. Then we have plant growth. Okay. So on the basis of effects of light on their vegetative developments, the plants can be divided into two categories. Very important for board examination. One is heliophytes. Okay. Or photophilous. Another one is, you can say heliophilus. You can call them only heliophilus. This can be called photophilus or heliophilus. Or I can call heliophilus one more thing. I remember correctly, shade intolerant I can call them. They are not able to take shade. So they are known as shade intolerant plants. It is the best way to describe them. Okay, which grow in strong light. Okay, means they do not grow in shade. Strong sunlight. Okay, they'll have their leaves oriented along the direction of the sunlight. Okay, to prevent them from photo bleaching. Okay, strong sunlight. The example is sunflower, pines, teak. Okay. And then the another one that you have is the opposite of that. Okay, they have heliophytes. You have now heliophobus, sciophytes. Number 2 is sciophytes / photophobus. Okay, they are photophilus. Here photophobus, like hydrophobic, hydrophilic, photon loving, photon hating. So photophobus. I can call them heliophobus. Helion again means, you can say, light. So heliophyte there, here, or heliophilus there, here, heliophobus. Okay, or I can call them shade tolerant. They love shade. So it's all conceptual only. Shade tolerant plants means which grow in lower light intensity. Okay, shade plants like spruce, herbs, shrubs. Okay, etc. Then we can come on to the seed germination issue. Seed germination. Once seed germination is done, so that is on the basis of response of seeds towards light. That is photoblastic response, it is also known as photoblastic response. Okay. So the plants are divided into 3 categories like that. Again, based upon how will the seeds react to light or equal to h nu, that is positive photoblastic, okay. Then you will be having negative photoblastic and non-photoblastic. So we have 3 non-photoblastic. Okay. So once we talk about positive, means whose seeds require light for their germination, that is positive photoblastic. They're dependent upon light, right? So example is viscum here. Negative means whose seeds germinate without light, that is in the absence of light, like onion, lily. Okay. Non-photoblastic means again like day neutral, like whose seeds germinate irrespective of light. Okay, you can compare it like that. It doesn't depend on photoperiod. It doesn't depend on, you can say, if you provide light, don't provide light, it doesn't depend. It will germinate like bean, maize, etc. So that is your light. Okay. Again, if you talk about stomatal opening, so we know that already the light favors the opening of stomata by promoting the, if you want to know the mechanism, okay, stomatal opening. This is asked in NEET examination. So I'm again reiterating it. So the light favors the opening of stomata indirectly. How indirectly? It has to start a signaling pathway. It promotes the malic acid formation. Okay, malate formation. Malic acid formation. Okay. But in case of xerophytic plants, xerophytes, what happens is light stimulates the closure of stomata, okay, which close during the day and open at night. Okay. Thus the opposite. Closure of stomata at daytime, meaning that I mean to say they will be closed during the daytime. That is known as scotoactive stomata. Comes in NEET examination. Scotoactive stomata. Okay. Means which will open at night. Okay. Then you have various phototropic movements like bending of shoots or plants, right? Phototropism. They are the indirect influence of light. Okay. Then in class 10th you might have studied about photonastic movement. Okay. So under phototropic you have geotropism, hydrotropism. Okay. So light means phototropism only. So again photonastic means you can say sleep movements. Okay. Leaves of oxalis are expanded horizontally during day and droop at night. Okay. So they can also control the circadian rhythms in plants. In plants also it is present. Okay. That is photonastic movement or sleep movements. In case of plants, without touch, that is a nastic movement. Without a touch of the stimuli, or not even touch, without being influenced on the direction. Okay, so it doesn't go towards the direction of the stimuli, it can go against the direction also. That is your nastic movement. Okay, so now if we talk about, you can say stratification, or directly we can talk about the thermal factor. Okay, so stratification in terrestrial communities is regulated again by light. I'm telling you it in short. So the dominant species, what they'll do, they'll occupy the highest canopy level to utilize maximum light. This you must put into consideration. So for utilizing maximum light, they occupy the highest canopy level. The dominant species occupy the highest canopy level. That is known as stratification. Stratum, okay, or strata in plural, stratification. Okay. So now we are coming to the thermal factor or temperature. Okay. Temperature. So temperature is the degree of hotness or coldness of a substance. Very easy. Okay. But it is the most important and critical ecological factor. Okay. Keep in mind it is the most important / critical ecological factor. Okay, which affects almost all the metabolic activities of an organism. You can just correlate if we have fever. Why do we have fever? IL-1, interleukin-1, which is a pro-inflammatory cytokine, is released by the immune cells after any kind of bacterial or viral or any kind of pathogenic infection that leads to the resetting of the hypothalamic clock. Okay, so it resets at a lower temperature. And then we start rubbing and shivering because that will generate friction and that will lead to us doing more work, and that work will lead to the generation of heat. So we are generating heat by shivering and rubbing. okay, and trembling, okay, and increase, you can say increase in the body temperature indirectly like that will prevent any biocatalyst activity or enzymatic activity, because enzymes are active only at certain body temperature, that is 98.5 degree Fahrenheit. If you go 9900, the enzymes will start getting deactivated, and even your ATP cannot be synthesized. Even if not, if an ATPase cannot work, complex 5, it is again an enzyme only, okay, transmembrane, it cannot work. Even ATP cannot be made, leave your trypsin, pepsin and other enzymes, okay. Even ATP cannot be produced, and you will be having cellular death or death due to the increase in body temperature, that is temperature, okay. So you need to take antipyretic drugs then, which will lower the temperature, like paracetamol, ibuprofen and henceforth, okay. So every physiological function has an optimum temperature, that is my meaning, that is what I want to convey to you, at which it shows the maximum metabolic rate, okay. So the vertical temperature gradient over the earth's surface has some value, and the vertical temperature gradient over the earth's surface is known as the lapse rate, okay. That's a new concept for you. The vertical temperature gradient, okay, over the earth's surface is called the lapse rate. Now this value comes equal to 6.5 degrees Celsius per 100, or sorry, per, you can say 1000 meter or 1 kilometer elevation. So 6.5 degrees Celsius per 1000 meter elevation, okay. Now change in temperature will be affected by factors like latitude, altitude, topography, vegetation, slope, okay. So temperature shows a great amount of variation, that is I wanted to convey, altitudinally and also latitudinally, okay. So the mean temperature of air changes by, you can say, 10 degree Fahrenheit, 10 degree Fahrenheit, if we switch from one latitude to the other, that is every latitude, 10 degree Fahrenheit at least, okay. So the temperature values are maximum at the equator, and because of this it would decrease gradually when we keep on jumping latitude and we reach the poles. Similarly, it also decreases from plains to mountain tops, that is a very common sense thing. Now you can say that it ranges from sub-zero levels at polar areas and high altitudes to, I can say, we have been able to detect and observe more than 50 degree of temperature also in deserts and also in the tropical deserts, specifically in summer seasons, in Sahara desert, okay, especially in the summer seasons. Even in Lucknow there is 46 to 48. This time it didn't cross 40 because of COVID-19 pandemic, less pollution and all. Now it has again reached to its normal level, okay, returned to its normal level, but it did decrease a lot, okay, when the lockdown was in full effect, okay. So it didn't cross 40, 42. This time I don't think it crossed 40 in Lucknow, but last year it did cross 45, 46, okay. So most of the organisms can survive therefore at a temperature range of 0 to 60 degrees Celsius, that is the average, okay, 0 to 60 degrees Celsius, okay, except certain bacteria, you can say cyanobacteria, there are exceptions. Some bacteria, then blue-green algae, this is cyanobacteria. Then there are some celled protozoans, some yeasts, okay, protozoans, then yeasts. So they cannot survive, Okay, and the bacteria and cyanobacteria are also there who can survive even in thermal spring, okay, like archaebacteria and deep sea hydrothermal vents, okay, which have more than 100 degree Celsius of temperature, and also in permafrost region. Hydrothermal vents also, thermophiles, archaebacteria, okay, and in the permafrost region also -30 to -50, they can stay at both the places. Okay, so they can do it, but we cannot, because again denaturation of enzymes will happen. Okay, so in the next class we'll talk about various thermal tolerance. Okay, like who can tolerate what range of temperature. Okay, mesothermal, microthermal, megathermal. Okay, then encystment. Then we'll be talking about the metabolism, then temperature based zone formation, zonation, tundra, then ice snow, then deciduous forest, grasslands, tropical. Okay, so and then effect of rainfall as you can say a very you can say detrimental and pivotal factor. Then aquatic adaptations in animals also. Okay, you have swim bladder, osmoregulation apparatus, okay, hypotonic urine. Okay. Then you have various rules. Okay, Allen's rule you have. Okay, so those rules are asked directly in board examination. Okay, so those we will be discussing in the next class. So I hope you are able to comprehend the lecture. If you have had any kind of doubt, you can directly post your query on my personal number. I will be replying as soon as possible. Okay then. Thanks a lot. Let's meet you in the next class.